Hi student, I am Dr. D.S. Thore. Now I share the screen of today's lecture in front of you. Please wait. Welcome to the 19th lecture of course number PATH-354. Title of this course, Diseases of Field and Horticultural Crops and their Management. Today we will discuss about diseases of banana. Okay, so in banana, some fungal, bacterial, and viral diseases are seen. So first, Panama wilt, Sigatoka leaf spot. These two diseases caused by the fungal disease. Moco wilt caused by the bacterial pathogens, that is Ralstonia solaniferum. And last disease, this is the most important disease, that is a bunchy top caused by the banana bunchy top virus. So see one by one. Okay, so first of all, we will getting information about Panama wilt. So this Panama wilt is also known as the fungal wilt. Okay, so this disease is widely spread throughout the Africa, Asia, Australia, West Indies, and some parts of the also India. Okay, so this disease was first discovered or found in Australia in 1818s. Okay, but it reached to the states, in particularly America. So in Panama, in 1950s, this disease most prominently seen on the banana crops. So it destroyed 40,000 hectares of the banana crops. Okay, so now recognized as a devastating disease or most important disease in Central America. Okay, so this is some ecological, these economic importance points behind this Panama wilt. Okay, so after the economic importance, we go to the next point, that is symptoms. Okay, so in Panama wilt, most conspicuous or most evident symptoms usually appears on the banana plants at least 5 months old plants. Okay, so under favorable conditions, the younger seedlings of the banana also suffer to this fungal pathogen and produce the symptoms. Okay, so after infection of the fungi to this banana, so initially symptoms are seen, majorly seen on the older leaf of the mature plants. Okay, means initially faint yellow streaks seen on the petiole portion as well as lowermost leaves of these plants. Okay, means affected leaf shows progressively yellowing symptoms. So these yellowing symptoms generally seen on the leaf of these plants. So these affected leaf or affected petioles, so uh, see these petiole portions. So these affected petioles and leaf generally break at this point and hang down along the pseudostems. Okay. But in the younger leaf, these symptoms generally seen, but the younger leaf remain erect and they also get affected under severe cases. Means main leaf or heart leaf remain upright in directions. Okay, then this fungus spread the from leaf to the petiole to the main pseudostems. Okay, means the discolorations appears firstly on the outer or oldest leaf sheath and extend up to the pseudostem. Okay, so you see this discoloration takes place within these pseudostems. Means fungus grows within this pseudostem. So finally block the vascular systems. Means the wilting of the these plants. Means affected plant gives finally odor smell. Means emit the odor smell from this affected pseudostem because this internal tissue is finally rotted. Okay, means affected plants may or may not be produces the bunches. Okay, suppose the bunches is formed on the affected plants, so these produced fruits are malformed or ripen prematurely or ripened fruits in bunches irregularly patterns. Okay, so finally these infections reach up to the root surface. Means root of the diseased rhizomes are frequently blacked in color and somewhat decay. Okay, so this is the symptoms develop on the leaf, pseudostem and fruits of this affected banana plants. Okay, so you see this initial infection seen on this 2 or 3 month old banana seedlings. Means the lowermost leaf firstly infected by the fungus and showing the yellow colors. Okay, so this is the whole plants to be wilted. Okay, somewhat in the severe infection, these affected pseudostems generally crackings. So this is a cracking portion seen on the pseudostems. Okay, so this is the affected banana plot due to the fusarium fungus. Okay, then we go to the causal organism, that is a fusarium oxysporum species pubins. This fungus majorly attack to banana and produces the fungal weight. Okay, so these pathogens produce the hypa and mycelia. So this hypa and mycelia grows into the host cell by inter and intracellularly. Means this fungus penetrates within this healthy one host tissue by forming the inter and intracellular mycelia. Okay, after that this fungus develop the spore fruits and this spore fruit is known as the sporodochia. Okay, so inside the sporodochia the conidia and conidiophore generally produced. Means in sporodochia two types of the conidia majorly produced. One is the macroconidia and microconidia. Okay, So you see this conidia. This conidia is known as the macroconidia. Okay, so these macroconidia are four-celled to eight-celled, and this macroconidia generally sickle shape. Okay, so 1, 2, 3, and 4 means four-celled up to eight-celled macroconidia generally produced within this Sporodocia spore fruits. Okay, then another conidia is known as the microconidia. So this microconidia is one-celled or two-celled. This is the one or two-celled. One or two-celled microconidia, and this conidia is always oval to kidney shape. Okay, means micro and macroconidia is always produced on these spore fruits, that is known as the sporodocia. Under unfavorable conditions, these mycelia develop the chlamydospores, and these chlamydospores produce singly or maybe in pair. Okay, so this is the resistant spores to desiccation and unfavorable environment conditions. And this chlamydospores survival for more than 30 years in soil after their host has been removed. Means in the absence of the host crop, this chlamydospore survival in soil more than 30 years. Okay, so this is the etiological parts of this Fusarium oxysporum forma species pubens. Okay, so you see these photographs. This is the macroconidia, this is the microconidia, and this is the chlamydospores. Okay, then we discuss about the perpetuation of this Fusarium oxysporum forma species pubens. Okay, means this pathogen survival for long duration in the rhizome portions. Or suppose this infected seedlings is used for planting purpose, so 100% this is produced. Or these healthy one seedlings may be planted in the infested plots having fusariums. So this plot contain the banana plants also suffer this disease. Okay, means spread of the disease from one plot to the another plot or one plant to the another plant through root contact. Means use of the diseased rhizomes or suckers for planting purpose, so initiate the initial infections. Okay, so already this is produced in the banana plants, so these pathogens spread through irrigation water or soil attached with the implements, shoes and vehicles. Okay, suppose heavy rainfall generally seen in particularly banana fields, so increase the spread of the pathogen, because these pathogens means these chlamydospores is always present in the soil. So these chlamydospores and infected plants having the spores may be disseminated from one plant to the another plant and reach to the long distance. Okay, then favorable conditions. This Panama wilt disease much severe when the soil generally used, that is the acidic soil. Means these banana plants grown in the acidic soil having the pH less than 7, so 100% disease is produced. Okay, then soil conditions like texture and structure is not properly or waterlogged conditions also initiate the disease. Okay, then this disease is also initiate when these rhizomes or stem portion infect to this insect, means this insect injures and form the wounds to growing the stem and root portion, or increase the population of the nematodes, that is a Rhodophilus similis. These nematodes also initiate and associate with the fusarium, so finally increase the Panama wilt. Okay, then last, that is a control measures against these Panama wilt. Okay, so first one, healthy planting material should be used or obtained from the disease-free areas. Then avoid the ill-drained soil and prefer slightly alkaline soil, because this fusarium disease initiate in the acidic soil. So farmer can grow these banana plants in somewhat neutral soil, or soil having the pH 7 to 7.5. Then flood following for 6 to 24 months, or crop rotation with the rice. Then dipping of suckers in carbendazim solution, that is 0.1% before planting, because this fusarium fungus may be survival in the sucker portion. So kill the spores inside these planting suckers by dipping in the carbendazim solution, or prepare the carbendazim solution, that is 0.1%, and these solutions generally drench around the root of the infected banana plants. Then also before the actual planting of the seedlings of the banana or suckers, so application of the lime, that is 1 to 2 kg per feet to the infected feeds after the chopping of the planting parts. Okay, then growing resistant varieties, that is a Basrai, Puan and Mungil. So this variety is somewhat tolerant to this fusarium wilt disease. Okay, so this is the information about the fungal wilt. This fungal wilt is also known as the Panama wilt. Panama wilt means why this fungal wilt is also known as the Panama wilt, because in 1950 this this wilt, this is majorly seen in the Panama states in America. So that's why near about 40,000 hectares land is totally destroyed due to this disease. So that's why this disease is also known as the Panama wilt. Okay, then second most important disease seen in the banana, this disease is known as the Moko wilt or bacterial wilt. Okay, means this is generally seen due to the bacteria. Okay, means this disease was first recorded in Guyana in 1940 in Moko plantain. So that's why this disease is known as the Moko disease. In India, it was first reported from West Bengal in 1968. Okay, means in India, Robusta and Puan variety are much or most susceptible variety for this disease in particularly Tamil Nadu states of the India. Okay, so why this disease is known as the Moko wilt or Moko disease? Because this bacterial wilt disease was first recorded in Guyana in 1840 in Moko plantain. So that's why this disease is known as the Moko wilt. Okay, Now we discuss about the symptoms of this Moko wilt. Okay, so external symptoms seen in the banana plant due to the bacteria, it's same in regarding the fusarium wilt. Only internal symptoms is differ than the fusarium wilt. Okay, means this affected banana plants is totally wilted and dries. Means initially yellowing leaf, this yellowing leaf just hang down to the pseudostems. Then this bacteria also infect to the pseudostem and fruits in bunches. Means only differ the internal symptoms. Okay, so the discoloration of vascular strands and blackening of the suckers generally seen in these photographs. So this is the infected suckers or stem portion of the banana due to the bacteria. Okay, means in fusarium wilt, no any bacterial ooze in comes out. But when the bacteria infect to these pseudostems, so almost this bacteria is releases from this infected pseudostems. Okay, means the vascular discolorations, that is pale yellow to dark brown or somewhat bluish black ooze, it comes out from this infected pseudostem and infected fruits formed in the bunches. This is the major difference regarding the fusarium wilt and bacterial wilt. Means this substance not secreted in the banana plants or pseudostems due to the fungal wilt. Okay, means grayish brown bacterial ooze is seen when the pseudostem of infected plant is cut transversely. Okay, means this firm brown bacterial ooze or yellowish or bluish bacterial ooze is also seen from this infected fruits formed in the bunches. So finally, the infected plants is totally dries and wilted. Okay, so this is the symptoms developed in banana when they infect by the bacteria. Okay, so keep in your mind about symptoms in fungal wilt and bacterial wilt. Okay, then causal organism, that is Ralstonia solanacearum or Pseudomonas solanacearum. This is the bacteria infect to the banana and produce the Moko wilt or bacterial wilt. Okay, so this bacteria is not harm the spores. This bacteria is always gram-negative reaction, rod shape and motile by 1 to 4 flagella. Okay, so this is the morphology of the Ralstonia solanaceae. Then we go to the perpetuations. Means primary infection takes place through the disease the plant suckers used for planting purpose. And secondary source of infection, that is this bacterial ooze mixed with the irrigation water or maybe stick on the surface of the implements, or these bacterial substances fed by the insect. So in this way, bacteria can move from infected plant to the healthy plant. So this healthy plant may be infected. Okay. Then control measures. So first control measures, that is eradicate the infected plants and suckers, killing in situ by application of the herbicides. Means regularly cut the suckers seen on the mother plants, or disinfestations of the tools, agricultural tools with the formaldehyde, that is using the 1:3 ratio of the soap solution, or maybe formaldehyde solutions. Then crop rotations with the sugarcane or rice at least 3 years. Then follow periods or flooding during the off seasons. Then fumigation of the infected pits or sites with methyl bromide or chlorophycline. Okay. Then expose the soil to sunlight during the hot dry weathers. The most important management point, that is use the healthy planting materials. Then spray systemic insecticide phosphimidon at the rate of 0.05%, because some insect acts as a vector to transmit the bacteria from infected plant to the healthy plant. So it is necessary firstly to kill the insect by applying the phosphimidon insecticides. Okay. Then somewhat also use these some antibiotic product, that is a streptomycin or other kasugamycin, to kill the bacteria. Okay. And last one, growing resistant varieties, that is monthon. This monthon variety is somewhat tolerant against these mucovade. Okay, so this is the information about mucovade or bacterial wilt. Okay, so in your semester end exams, one questions asked, that is write down the differentiate between fungal wilt and bacterial wilt. So you can write the some points to differentiate the fungal wilt and bacterial wilt. Okay. Then third most important disease, that is a Sigatoka leaf spots. So this is the most common and seen in the all the variety of the banana. Okay, so this disease was first observed in Java countries in 1902. Okay, nowadays this disease is the most common disease seen in India, especially Assam, Tamil Nadu, Karnataka, Maharashtra, and Andhra Pradesh. So significantly yield losses generally seen in banana due to the Sigatoka leaf spot. That is a 10 to 30%. Okay, so this is the most important disease. Then we see the which type of the symptom produced by the Sigatoka leaf spot pathogen in the bananas. Okay, so in infected banana plants, this initially this symptom generally seen on the third or fourth leaf from the top of the plants on the banana. Means initially this infection seen on the third leaf or fourth leaf of the banana. Means this infection majorly seen on the middle parts as well as the lowermost part of these banana plants. So initially small spindle shape spots seen on the